where we identified some novel underlying genetic factors. I don't have anything to disclose. So, as you may know, heavy menstrual bleeding is defined as the pathologic state as excessive or prolonged menstrual blood loss in excess of 80 milliliters per menstrual cycle. This occurs in about 1/3 of adolescent females, and it actually accounts for 2/3 of all hysterectomies. 50% of those with hysterectomies from HMB do not have their underlying cause identified. Quality of life can be significantly affected by heavy menstrual bleeding in the form of significant iron deficiency anemia, psychological distress, missed school, limitations on activities, and prolonged periods of required rest. The prevalence of bleeding disorders in heavy menstrual bleeding patients, as you may imagine, is higher than in the general population. For example, the prevalence of von Willebrand disease is 0.1% of the general population, but 13% in patients with heavy menstrual bleeding. More recently, low VWF levels, independent of a diagnosis of a bleeding disorder, have also been shown to be a significant risk factor for heavy menstrual bleeding. And to date, there have been no next-generation sequencing studies performed to determine genetic hemostatic risk factors for heavy menstrual bleeding, which might identify novel loci. There have been several genome-wide association studies of von Willebrand factor antigen levels, as well as number and volume of various cell types in the blood. But these types of studies do not detect association with rare variants. So 10 tertiary care centers in North America with expertise in hemostasis and that provided care for adolescents with heavy menstrual bleeding and were members of the Foundation for Women and Girls with Blood Disorders participated in this study. All subjects met the following eligibility criteria. They were all under 21 years old when diagnosed with heavy menstrual bleeding, as defined by a pictorial blood assessment chart or PBAC score of greater than 100, and low VWF as defined by having 2 or more values of VWF activity greater than 30 and less than 50 international units per deciliter. 86 patients had sufficient blood samples for whole exome sequencing. And although it might be expected that such a study would find an excess of rare and pathogenic variants in the VWF gene in this population of patients with low VWF, by taking an agnostic approach such as whole exome sequencing rather than a candidate gene approach, other genetic patterns can emerge. So we had exome sequence data for our 86 unrelated probands, and we had 660 unrelated in-house controls for whom we also had whole exome data that were all generated at the McDonald Genome Institute at Washington University. We aligned and joint called the exome data using the Sention software package. All of the annotation was performed using Anavar. We used standard quality controls for things like read depth, genotype quality, and allele balance in order to filter out poorly called variants. Principal components were calculated using Eigenstrat. Principal components from whole genome-wide single nucleotide polymorphism data of all common, meaning greater minor allele frequency of greater than 5% were used. We did not remove any cases because principal components confirmed self-reported race. Principal components are sort of data reduction metrics that are measurements of similarities between individuals derived from their genetic information. So the first few PCs, which are orthogonal, which basically means statistically independent variables, essentially represent the largest inter-individual differences derived from the data. And these are almost always based on ethnicity. So for example, in the case of samples, including different continental groups, people from different continents, PCs often separate those of Asian, African, and European descent. If only one continental group is included in the sample, then these will represent smaller changes representative of ethnic groups within that continent. And then the standard cutoff for genome-wide significance of 5 × 10⁻⁸ was used. We were reasonably able to exclude patients with heavy menstrual bleeding and low VWF from our control cohort, even though it wasn't ascertained specifically for that, because the estimate of the joint probability of low VWF associated heavy menstrual bleeding in the general population is likely around 1%, given that the prevalence of low VWF is around 2.5%, and the prevalence of heavy menstrual bleeding is 35%. So why do we care about rare variants? Well, the majority of genetic variation is very rare, as you can see on this graph of the site frequency spectrum. So in particular, the non-synonymous variants, which are in black, you can see are quite rare, and they have a disproportionate role in disease. So we utilized gene burden analysis, which is a technique that improves our power to detect rare variant associations. So as you can see here, each case and each control has a different rare variant in the same gene, which on their own would not be significant. But when the minor allele frequencies are collapsed across the gene, then we become able to detect significant associations between the frequencies of these variants in cases and controls. As you can see in these two examples, in example A, you collapse the rare variants in cases and controls, and then the difference is now statistically significant. And same thing for gene B. So this is a pretty powerful approach. So to quantify the enrichment of rare, non-synonymous, or splice site variants, as well as ClinVar pathogenic variants in heavy menstrual bleeding cases compared with controls for each gene, we did this. We collapsed the minor allele frequency of variants in cases and controls using Fisher's exact test, using only the filtered variants that I discussed earlier. And the variants were, like I said, filtered for things like rareness and quality. Because rare, likely pathogenic variants are necessarily enriched for causal variants, we first looked at the rare stop gain and stop loss, as well as ClinVar pathogenic variants in our sample. And among the 86 patients, we observed 4 who had rare stop gain or stop loss mutations in genes associated with bleeding or hematological disease. The patient that we found with a stop gain mutation in VWF had a frequency of this allele in NOMAD of nearly 0. So there was only 1 instance of this observed in NOMAD. This variant is not listed in ClinVar, but is likely to be the cause of the heavy menstrual bleeding in this individual, as they're also likely to have von Willebrand disease as a result of this mutation. So in controls, we only saw 1 rare stop gain mutation, and this difference was statistically significant. The variants that we found in these patients in the cases are known to cause platelet glycoprotein 4 deficiency, that was CD36 gene, von Willebrand disease, bleeding disorder, platelet type 11 in GP6, and hereditary coproporphyria in CPO. Next, we pooled all rare, meaning less than 1% in nomad, ClinVar pathogenic variants that were present in the cases that were in genes known to cause a dominant or a recessive form of anemia or a disease that had anemia as a major symptom. And we found that there was a statistically significant difference between cases and controls in the number of these variants present in each group. Cases had 9.3% and controls had 4.2% for a p-value of 0.04. This is the table of all the genes that we've found one of these variants in the cases. The disease association of the gene, as well as the inheritance mode for the disease, are also listed. We also performed the same comparison in genes involved in hemostasis and platelet function. I don't have a table for this, but we found that 4.6% of cases and 1.4% of controls had rare ClinVar pathogenic variants in these genes. And this difference was slightly statistically significant, but the association was driven by genes or by variants in cases that had these variants in VWF, factor 2, and LYST. The variant in factor 2 that was found is known to be associated with prothrombin-related thrombophilia, and the variant in LIST has been shown to be associated with Chidiac-Higashi syndrome, and coagulation defects are one of the known symptoms of Chidiac-Higashi syndrome. So next we did common SNP analyses. We tested for association with all common SNPs present in the exome and the genome data. So 5% in nomad. Greater than 5% or greater is considered common. 3 common SNPs in linkage disequilibrium. In or downstream of the gene FERMT2, Fermitin family member 2 achieved genome-wide significance in this analysis. And interestingly, although the minor allele frequency of these SNPs was around 10% in the non-Finnish European population of nomad, we found these SNPs present in 24% of our cases and only 6% of our controls. And maybe more interestingly, FERMT2 encodes for a cytoskeletal protein that is a crucial regulator of integrin function. So it's known to regulate hemostasis and is required for things like angiogenesis and blood vessel homeostasis. And additionally, we looked and these SNPs that were found to be associated with heavy menstrual bleeding in this gene are also expression quantitative trait loci for FERMT2, according to Genotype Tissue Expression Project. So this is just a quartile plot of the p-values for the common SNPs. And this shows the relationship between expected p-value on the x-axis and the observed p-value distribution on the y-axis. So deviations from a slope of 1, which is the red line, the diagonal red line, indicate p-value inflation or deflation, and specific points with a higher p-value than expected likely indicate real association. So for example, the 3 SNPs in FERMT2 that are circled that surpassed the genome-wide significance, which is denoted by the dotted red line. So this is a Manhattan plot, which gets its name from its similarity to the Manhattan skyline, a profile of skyscrapers, and then the lower level buildings. And in genome-wide association Manhattan plots, genomic coordinates are displayed along the x-axis. So each dot is SNP, and then the negative log of the association p-value for each SNP is on the y-axis. So because the strongest associations would have the smallest p-values, their negative log scores will be the greatest. And here again, the red line indicates genome-wide significance, and the blue line indicates exome-wide significance. So as you can see, in addition to FIRM-T2, which was genome-wide significant, as we discussed, we also discovered SNPs in 3 additional genes with p-values that surpassed exome-wide significance. The first one is EBAG9, which is estrogen receptor binding site associated antigen 9. And this is mainly produced by macrophages in hematopoietic tissue and has a crucial role in controlling erythropoiesis. The next one is TTC18, which is also known as CFAP70, cilia and flagella associated protein 70. And this is expressed in the cilia of glandular cells in the fallopian tube, endometrium, and respiratory tract. And then lastly, TCN1, which is transcobalamin 1. And this is highly expressed in granulocytes, which have been identified as critical determinants of uterine bleeding and tissue remodeling in a mouse menstruation model. So some interesting, potentially biological candidates that make biological sense here. We then compared the genome-wide frequency of rare coding variants in our heavy menstrual bleeding cases and controls. An increased burden of rare variants that surpassed exome-wide significance was seen in the 10 genes here in this table. Our top hit, which is CFB complement factor B, encodes for a component of the alternative pathway of complement activation. And rare variants in this gene have been associated with atypical hemolytic uremic syndrome, which is characterized by microangiopathic hemolytic anemia and thrombocytopenia. And our second most significant gene was DNase2, deoxyribonuclease 2 lysosomal. And this encodes a protein which mediates the breakdown of DNA during erythropoiesis and apoptosis. And it's been shown that patients with complete loss of function of this gene have severe anemia and thrombocytopenia. So again, some top hits that make potential biologic sense, which is always encouraging. And next, we took the gene burden analysis a step farther and performed gene set burden analysis, whereby variants are first collapsed at the gene level and then by their gene ontology term membership. And for this analysis, we used non-synonymous and splice site variants that were less than 1% in nomad, so the rare variants. And then, as I said, the numbers of variants within the groups of genes were summed based on their membership within a given gene ontology gene set. And then this was used as the dependent variable in a linear regression. So for this analysis, the exome-wide p-value cutoff was used in the gene burden analysis because this is the number of independent tests, right? So you take the maximum significant p-value cutoff, which is 0.05, and then you divide it by the number of tests. And so there's around 20,000 genes. So that's how you get the exome-wide p-value cutoff. And additionally, for our disease-associated gene set burden analyses, we created a disease-defined set of genes that were involved in anemia using the online Mendelian inheritance in man, OMIM, and UniProt disease descriptions by searching for the term anemia in the disease description. And then for the bleeding disorder disease defined set, we searched the same databases for the terms bleeding, platelet, and hemostasis. And so here you can see that these gene sets here were statistically significant between cases and controls. Oxygen transport activity, hemoglobin complex, platelet degranulation, positive regulation of erythrocyte differentiation, and platelet alpha granule lumen. To ensure that these were not just false positives, we also examined 5 control gene sets in cases and controls in the same manner by using the gene ontology terms. And for the controls, we used regulation of lipid metabolic processes, breast and ovarian cancer, microvascular complications from diabetes, epilepsy, and autism. And none of these were significant between cases and controls, which was reassuring that our results were real. So I would like to recap what we've discussed so far. So firstly, we've shown, we've replicated actually what other investigators have noted in patients with heavy menstrual bleeding, which is this enrichment of pathogenic variants in genes that are known to cause bleeding disorders. And we even saw one rare, never before seen and no stop gain mutation in VWF. And this enrichment of rare ClinVar pathogenic variants, also in hemostasis and platelet function genes that I mentioned in cases and controls. Next, we find that heavy menstrual bleeding is associated with common variants in hemostasis genes. So the common variant analysis showed the association with FIRM-T2. This gene is also called KIND2, kindlin2, and it is involved in regulation of hemostasis, angiogenesis, and blood vessel homeostasis. And in animal models, partial reduction of this gene in mice resulted in fewer blood vessels. And then the existing vessels lacked smooth muscle cells and were thinner and shorter than normal. And these knockdown mice had prolonged bleeding and vascular occlusion times because of the suppression of platelet aggregation from elevated expression of 2 enzymes on endothelial cells. And complete knockout mice of this gene are embryonic lethal. We also found that heavy menstrual bleeding is associated with rare variants in anemia genes. So iron deficiency anemia is a common but underappreciated complication in adolescents with heavy menstrual bleeding. And the variants that we identified could be causing less severe or potentially incomplete penetrant forms of anemia. We observed significantly more rare ClinVar pathogenic variants in 8 genes associated with varying types of anemias in cases than in controls. A subset of these genes cause dominant forms of anemia, so this suggests that it's possible that at least a subset of the patients with these rare pathogenic variants could actually have undiagnosed disease. And in these patients, the anemia could precede the heavy menstrual bleeding, so would be therefore exacerbated by the blood loss. And patients with variants in the genes that cause recessive and forms of inherited anemias, like I said, could have milder forms of the disease as they would have one functional copy of the gene. And then we saw that the 2 most significant genes from the gene burden analysis were directly linked to anemia. So those rare missense mutations in CFB were associated with atypical hemolytic uremic syndrome. And the mutations in DNase2 have been associated with a loss of endonuclease activity, which causes severe neonatal anemia and thrombocytopenia. And then lastly, we showed that heavy menstrual bleeding is associated with common variants in anemia genes. So the common variant analysis, as you recall, revealed that association with EBAG9, which induces apoptosis in normal human erythroid progenitor cells. And erythropoiesis is in part negatively regulated by macrophages through production of this gene, which contributes to the pathogenesis of anemia. And so from here on is not in the manuscript. This is work that has been done since the publication of the manuscript. So since then, we've looked into replicating our findings in the UK Biobank dataset. The UK Biobank is a collection of 500,000 individuals with paired genetic and phenotypic information. It's a very powerful dataset. Most studies using this dataset, however, only examine individuals of European ancestry. But this group from Harvard recently performed and made publicly available a multi-ancestry analysis of basically every phenotype in the UK Biobank across all of the 6 continental ancestry groups that they have, for a total of over 16,000 genome-wide association studies, which are just freely available for download. So here are the p-values for any results from our analyses that we found that were also significant when analyzing the UK Biobank genetic data. We see a slightly significant replication of DNase2 in the gene burden analysis in the UK Biobank data, as well as TBL1X. And the protein here encoded by FRMT3 has a key role also in regulation of hemostasis and thrombosis, and might help maintain the membrane skeleton of erythrocytes. So you recall in our GWAS, we found that FRMT2 significant. However, here we find FRMT3, but they do have sort of similar mechanisms. So this could be interesting. And in our GWAS of the UK Biobank data, there was one SNP in TCN that was TCN1 that was significant here at the bottom. And this gene, as you remember, was found to have SNPs that were exome-wide significant in the GWAS of our own data. So I should say that this data here is only based on 200,000 of the 500,000 UK Biobank participants, because this is what we personally have access to the genetic data for. But this here is the publicly available GWAS data from the Pan-UK Biobank that I mentioned for several menstrual bleeding phenotypes. So this is all 500,000 people in the UK Biobank. And here you can see that the data is broken down into cases and controls for these various menstrual bleeding phenotypes, and it's also broken down by population. So you can see that most of the people are European ancestry in blue, but they also have Central and South Asian ancestry in orange. African is the purple, East Asian is the green, Middle Eastern is the pink, which there's very few of, and then admixed American ancestry is the red, which is also a very small proportion here. And here are the results from the Pan-UK Biobank public GWAS data for excessive and frequent menstrual bleeding. And we see significant peaks at FSHB, which is follicle stimulating hormone subunit beta, which interestingly induces egg and sperm production. And F5, which is coagulation factor 5, whose activated protein is actually a cofactor that participates with activated coagulation factor 10 to activate prothrombin to thrombin. And defects in this gene are known to result in either autosomal recessive hemorrhagic diathesis, or an autosomal dominant form of thrombophilia. So again, each of these make some biologic sense. And you see here that even when combining multiple menstruation phenotypes together, that these genes remain the two most significant, FSHB and factor V. The others that are listed don't have an obvious biological mechanism, but that does not mean the association is not real. Often in GWAS, it is not clear how a gene is affecting a disease, and so this is why things like animal models are so important. So for our conclusions, this is the first whole exome sequencing results of heavy menstrual bleeding patients, as well as comparison to control exomes. However, in the future, we would like to have phenotyped controls of our own and not just rely on UK Biobank or other data sets such as those. We observed an excess of rare non-synonymous variants in cases in genes involving several bleeding disorders, as well as an excess of these variants in genes causing anemias and disorders with anemia as a symptom. And then in our UK Biobank analyses, the heavy menstrual bleeding cases in the UK Biobank were not specifically ascertained on low VWF and a p-value score of greater than 100, like our cases were, and is also obviously a much larger sample size, and has no rare variant information because GWAS by definition are common variant analyses. So this could possibly explain the differences that we see between the analysis of our cases and the analysis of the cases in UK Biobank. So next, there's really a need for a functionalization of any potentially interesting variants. So with that, I would like to thank Jorge de Paula, who is my mentor at Washington University School of Medicine, Gabe Haller and Christina Gurnett for letting me use their control samples. And then our collaborators, Dr. Peter Coides at Rochester, Lakshmi Srivis at UT Houston, and Charles Menard at Baylor, as well as the rest of our co-authors and our funding. And with that, I would like to take questions. Thank you.
All right, we will go ahead and get started with our questions. Let's see. Okay, I do have a few for you. Let me go ahead and get started with the first one, which is, how can your findings enhance clinical care?
Yeah, so it's hard, right, when you have only 86 patients. It's not a lot of power, but I do think that the most interesting finding is actually the anemia, and that some of these patients could have been living with anemia and not known it until they got their period, because they would have had a rare variant in a gene that causes a dominant form of anemia. So I think if they could have knowledge of that beforehand, then it could prepare them at least for the bleeding that happens when you have your menstrual bleeding. So I would say I thought that was our most interesting finding, and it's I don't know that it helps patients so much after the fact, but it's more just knowledge for the knowledge of what you might have to deal with in the future, as well as who wouldn't want to know that they had anemia, right? It's just good to know.
Right, absolutely. So that kind of brings me to my second question, which was, there were several interesting findings from your presentation. It's a two-part question. Can you elaborate more on your second finding? But I guess you also answered the second part, which was which finding was the most interesting and exciting for you, which was the anemia one.
So the first part of it was
elaborate on the second finding.
Yes.
Which one was the second one?
Oh, gee, let me see from the video.
No. Like the one that says number 2?
Number 2, yes, yeah.
Okay, which one is that? I'm sorry, your slides are not going back.
Let me go ahead and try to pull that up real quick. We'll go ahead, I'll look for that and we'll come back to this question. But so do you see the potential for screening through genetic testing that could result in diagnosis and treatment down the line for HMB?
Yeah, I mean, I do. There, like I said, there's already a paper out that, you expect there to be an increased proportion of patients with heavy menstrual bleeding that's caused by, an underlying bleeding disorder like von Willebrand disease or hemophilia. But you don't necessarily know this, but even a greater proportion, at least in our data, was explained by these very uncommon variants in anemia genes. And I think that, if we can replicate the FIRM-T2 finding, if we can measure expression of that, because the SNPs that were associated with that particular gene were expression quantitative trait loci. So if people that have heavy menstrual bleeding have significantly different expression of that gene than controls, then that would be big support for that finding. So I think that would, just a lot replication and strengthening your belief in the data through various avenues is ultimately what I think leads to confidence to help patients. More knowledge is needed. I hope that answered the question.
Sure. Yeah. Okay, so the second finding that they'd like for you to elaborate a little bit more on was heavy menstrual bleeding is associated with common variants in hemostasis genes.
Yeah, that one I didn't put the table. I'm sorry. So this is sort of what I was talking about, was with that we replicated the finding that others had shown that there was an excess of variants in genes that are known to cause bleeding disorders, and we did see that in our patients, but that difference was less statistically significant than the difference between cases and controls with regard to the variants in the anemia genes, which is sort of what I was saying. But I didn't have a table for that. I'm sorry.
No,
but it was only 4 variants, which is why I just
yeah, absolutely. I have my last question, so I'm going to throw it out there that if anyone else has any additional questions, go ahead and throw them in the Q&A feature at the bottom of your screen, or you can send them directly to me in the chat. But your final questions are, what are your next steps in this research and how will you build on your findings?
Yeah, I've actually was just talking with our collaborators this week, and they want to do some functionalization too. I think that the FIRM-T2 finding is an easy place to start, because you just need to measure difference in expression, but also some of the exome-wide significant findings there would also be good candidates. And we also would like to get our own controls, as I said, like our own phenotyped controls, because the controls that I was using were, ascertained for other projects that are completely unrelated to hematology in any way. So for the purposes of this analysis, are controls, but we don't have the PBAC score for them. We don't have their antigen levels. So UK Biobank is great, but if it's a lot smoother if you have controls that you did in the same way that you ran your cases, so that you can be certain that everything was done exactly the same and on the same platform and etc. So we would like to phenotype some of our own controls.
That sounds very exciting. Okay, what were the greatest challenges in your research and how did you mitigate these?
Greatest challenges, well, having only 86 people is kind of a challenge. You know, sometimes in human genetics, they don't fund your grants unless you have like 1,000 people or more. So I just I knew I had to keep the focus on the rare variants, mostly because of otherwise it would have been hard to find much in so few people. So I think just trying all the different types of rare variant analyses that I could think of. You just want to get the most out of your data because, you know, it doesn't cost a small amount to sequence that many people, although much less than it used to, of course. But you know, you just want to, since it was like up to me, I was the analysis person on the project, so I just had all these people looking at me like, what's the answer? Tell us the answer. So just trying to maximize the potential of the data, I think, was the hardest.
Sure. All righty. Well, I'd like to thank you once again, Dr. Sadler, for taking the time to join us this afternoon. As always, we appreciate your time and expertise. And I'd also like to thank each one of you viewers for joining us. Please note that this recorded webinar will be available on Friday, April 15th at hemophilia.org under the Events tab with all of our archived webinars. Also available in the Events tab is our upcoming schedule for our weekly Wednesday webinar series. Thank you for joining us this afternoon.
Thanks for having me.
Yeah, of course. Well, I hope you guys have a wonderful week. Thank you once again.
Thanks.